All right, this podcast is about DNA and its structure. We know that DNA is the genetic material. It is what creates you, what allows you to be functioning living thing, and DNA is common to all life on Earth. It is all built the same way, whether it's in a bacterium or a person. It is the unifying one of the unifying molecules of life, of living things. DNA can do two really important things. It can copy itself so that we can pass on our genetic information, our traits, to our offspring. And of course, it directs the construction and functioning of our bodies. And when I say our bodies, I mean the body of any living thing. It does that by creating proteins. When you look in the mirror, you're made of protein. That's what you see. Your body is basically protein. DNA is the instructions for building all of that protein. Okay? So it copies itself so you can pass on the instructions to your children, and it creates you and builds all of your proteins. So, DNA structure, okay? It is a nucleic acid. It is the most famous nucleic acid. There's another one, RNA. Please remember that nucleic acids are polymers. Let's not forget that term. Many monomers linked together. So, the monomers that make up DNA are nucleotides, okay? Nucleotides. So, in this case, every circle here is a monomer, if you'll remember from earlier in the year. And so if we're talking a DNA molecule, those are all nucleotides. Now, each of those circles, each of those nucleotides, is in fact made of three smaller parts. So it's almost as though we divide each of these into three parts. Every nucleotide is made of three parts. A sugar, a nitrogenous base, and a phosphate group. Okay? So you need to be able to kind of recognize this generic thing as a nucleotide. This whole thing is a nucleotide. The base may vary between nucleotides, between one nucleotide and the next. The base may change, but the phosphate and the sugar will always be the same. They're always the same. Okay? The sugar is a 5-carbon sugar instead of 6, like glucose is a 6-carbon sugar. This is a 5-carbon sugar called a pentose. It is more specifically called deoxyribose, hence DNA, deoxyribonucleic acid. In RNA, the sugar is a little bit different. It's ribose, but it's really trivial for us, that difference. And you need to understand just how the molecule is structured in diagrams. Usually you see this ring shape, and at every bend it is just understood there is a carbon there. So we start at this oxygen atom, and by convention the carbons are numbered. So we start here and we go clockwise to the first carbon. It is called the 1', then the second is the 2', 3', 4', and then this one that's sticking off is the 5'. This is important. You need to know about this carbon being the 3' carbon and this carbon being the 5' carbon. Okay? Please keep that in mind. The phosphate group is always attached to the 5' carbon. So right here, that one sticking up is always going to have the phosphate group on it. Okay? Now, those bases that I said could vary, okay? There are 2 families of bases. They are called purines, as we see here, and they are all 2-ring molecules. See here, here's a ring, 2 rings, 2 rings, purines. And the pyrimidines, the other family, is a 1-ring shape, okay? This seems very trivial. The old exam, though, used to expect you to know it. I don't know if this new exam will really care, but that's how that works, okay? The pyrimidines are cytosine and thymine and uracil, which we'll talk about later. The purines are adenine and guanine. In DNA, just remember C and T and A and G. Sorry, I'm writing at a weird angle here. Now, please remember from your first year biology, C always pairs up with G. A always pairs up with T. So it's always a purine pairing with a pyrimidine, a big and a small, okay? And I have a terrible time remembering these. I can remember by shape. Purines, the small word is the larger molecule. The big word is the smaller single ring molecule. But what is most critical for you, remember, is how they are paired. C to G, A to T, okay? Now, in order to make the big polymer, nucleotides have to be linked together, okay? Covalent bonds, please remember those nice strong covalent bonds are what link one nucleotide to the next to make a long chain, okay? Remember, we're always making long chains. We're making a big polymer here out of our monomers. So these bonds here, notice this is the 1, 2, 3, 4, 5. This is the 3' carbon attaching to the phosphate group on the 5' carbon. So 3 and 5' carbons are the ones that are going to connect in these long chains, making these covalent bonds, okay? The type of covalent bond is called a phosphodiester bond. If you'll remember back when we were doing biochemistry, we had glycosidic linkages connecting carbohydrate monomers. We had ester bonds linking parts of lipids and peptide bonds. Well, this is just another one of those. They're all covalent, and that should be what you need to remember. Okay? So, the sugar and the phosphate molecules are making up the backbone of the nucleic acid. In other words, the sugar, phosphate, sugar, phosphate, sugar. Whoops. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That's what's important in linking up a strong backbone. Remember, these bases may vary a bit, okay? All right. Please remember that the DNA molecule is constructed as a double helix, two nucleic acid ribbons spiraling together around an imaginary axis, okay? If you look, here is one of those ribbons, and here is the other here. And if you notice, they are upside down relative to each other. These are the covalent bonds here, linking them together, one to the other in a long, strong chain. But notice there are nice weak hydrogen bonds that link the bases in the middle. Okay, that's not by accident, because when we are working with this molecule, when the cell is working with this molecule, it has to be able to unzip itself down the middle, and so those nice weak hydrogen bonds allow that to happen. Please also notice the A to T, C to G, C to G, A to T. A always pairs with T, C always pairs with G. Okay? All right. Okay. The hydrogen bonds, there's the strong covalent bonds, the weak hydrogen bonds. Okay. And we've already talked about C to T, A to G, so we don't really need that. The word complementarity, you do need that because that is the word used to describe the A to T, C to G nature. If we have A on this side of the ribbon, we know that T is going to be on this other side. If we have C, we know there's going to be a G. Complementary, that concept is really important with DNA. A is complementary to T, C is complementary to G. That is important because we know that each is a predictable counterpart of the other, okay? And that's really important in the fact that DNA can copy itself and that we can understand it, okay? So There's a bit here about discovery of the structure of DNA. If we have time, we'll catch that in another podcast. But that's it for this one. We'll talk about replication next. So thanks for watching, and we'll catch you in the next podcast.